So good morning, everyone. I'm Michael Kelly, the Chief Scientific Officer at CureDuchenne, and thank you for joining us today with Avidity Biosciences for the first presentation to the Duchenne community on the data from the EXPLORE44 clinical study. Now, just a few housekeeping points before we get started. We've provided an opportunity for Q&A at the end of the webinar. You can submit questions at any time by clicking on the Q&A tab at the bottom of the screen. Any questions we don't get to today, we'll work with Avidity and get a response after the webinar is finished. This webinar is being recorded and will be available afterwards on our website. CureDuchenne was an early investor in Avidity, and our missions were aligned to help accelerate the development of novel muscle-targeted exon skipping drugs that targeted all muscle groups and improved the efficiency of exon skipping. I suspect that many of you on the call this morning will have seen the company's press release a couple of days ago. The results were remarkable, and in my opinion, highly suggestive of a potential significant clinical benefit for Duchenne patients taking Dalzota. To talk about the data, I'm joined this morning by Sarah Boyce, the President and CEO of Avidity Biosciences, Dr. Diana Castro, the founder and director of the Neurology and Neuromuscular Care Center in Dallas, Texas, and Hussam Yunus, the DMD Global Program Head at Avidity Bio. So without further ado, I'll hand it over to Sarah to get things moving.
Thank you, Michael, and thank you for joining us this morning and this afternoon for those of you who are on the East Coast for the presentation and discussion on the initial data from the EXPLORE44 clinical trial. This trial is an evaluation of Dalzota, which you may also see as having been referred to as AOC 1044, for the treatment of individuals with Duchenne's muscular dystrophy amenable to exon 44 skipping, also referred to as DMD 44. And if we can go to the next slide, please. Avidity is a publicly traded company, and as part of being a publicly traded company, we always have to make sure that everyone is aware that during a course of a presentation, we will be making what's called forward-looking statements. On our website, you'll see a full paragraph that explains all of these in detail. But essentially what they are is forward-looking statements is a term used by law that allows us to say things based on what we know or our current beliefs, even though they are not guaranteed and they can change due to factors that are within and beyond our control. This presentation does contain what's called forward-looking statements, including but not limited to the categorization of the initial Dalzota data, the EXPLORE44 trial designs and goals, our expectations for regulatory interactions related to Dalzota, or in relation to EXPLORE44. In terms of now moving on to our vision as a company from when we founded it to where we are today and going forward is pretty simple. We're looking to profoundly improve people's lives by revolutionizing the delivery of RNA therapeutics. RNA therapeutics as a class of drug, as we all know, have been around for a while, are very well established. But one of the fundamental challenges has been that of delivery, and in particular, delivery of an RNA, or in this case for DMD, PMOs to muscle cells. And you'll see how we have really sort of made enormous strides in solving that challenge with our technology. And if we can move to the next slide, please. So joining me today is, as Michael explained, Dr. Diana Castro. She's going to go through the preliminary data assessment. And then from there, Hussam will talk around, our future plans, as well as both for Dalzota as well as for other potential exons as well. And then most importantly, we'll get into a Q&A session. So we can go to the next slide, please. So first and foremost, we're here today because of you and together with you. We have had a deep partnership with the community from the very beginning. We're incredibly grateful for CureDuchenne as being one of our early financiers, and from that financing was very important when we were a small company with big dreams, but no real data to say if those dreams could be realized or not at that point. So we're very grateful for CureDuchenne for their partnership from the very beginning. I also want to make sure that it's important to thank everyone who participated in the trial, both and are continuing to do so, the investigators, the patients themselves, the families. We know it's a huge undertaking to participate in clinical trials. Now, one of the things at the beginning, you've heard us talk about Dalzota. So formally, it's AOC 1044. There's a generic name, which is a very long name that's given to you actually by the World Health Organization, which is Delpacibart. The Delpacibart refers to the antibody, and zotadicerin refers to the PMO or the oligo. So the four kind of tails that you see that are green on this slide. Given that Delpacibart to zotadicerin is a mouthful, we've shortened it to Dalzota. So that's what we'll use during the rest of this presentation. If we can go to moving to the next slide. So what we're going to share with you today is the initial data, an update on the next phase of our program, the importance of partnership, a reflection on the important contributions of study participants to improve care and advance treatment options for DMD. And I hope that you also get a sense of our urgency and our commitment, our deep commitment to improve lives of those people who are impacted by DMD. And clearly the data with Dalzota is the start of that. With that, I'm going to hand it over to Dr. Castro, who's going to take you through the data. Over to you.
Thank you, Sarah, and good morning, everybody. Thank you so much for having me here presenting this data. I love these opportunities because every step we take forward is a big step for us, for all our patients. And thank you for CureDuchenne always for having my back. So today, like Sarah said, we'll talk about EXPLORE44 preliminary data assessment. I'm so glad they shortened the name because I don't think there was a way that I was going to be able to pronounce the whole thing. So if you can move forward, next one. So it's important to understand first the study design and what we're talking about today. So there were two parts of the study. There was a part A for healthy volunteers and a part B for participants with Duchenne muscular dystrophy who are amenable to exon 44 skipping. As you know, right now we have other products that are for other type of deletions, for example, for exon 51 amenable, for exon 45, for exon 53. This is a completely different group, and obviously it's a new type of product, new type of information. So the one we're going to discuss today is actually B1. Is the first part with the lower dose of 5 milligrams per kilogram. This is a 7.5-month treatment and observation. And this is what we're going to talk today about. If you can go to the next one. So the overview and objectives. What is important also is understanding how did it work. So the patients are receiving multiple doses, total of 24 patients between the ages of 7 to 27 years of age. There were patients that were ambulatory and non-ambulatory, which is extremely important whenever we get, hopefully down the road, hopefully one day, approvals where we include patients that are non-ambulatory as well. The biopsies were performed in all cohorts, and the participants are eligible to roll over into the extension study. The primary objectives are safety and tolerability. Always when we do these first studies, we're looking always to safety and tolerability of the multiple dose of the product, of the drug that we're investigating. The secondary objectives are what we call pharmacokinetics, pharmacodynamics. It only refers to the distribution of the medication and some of the good effects that or effects that will have in the body, and dystrophin protein levels. And the key exploratory objectives are looking into measure of muscle function, patient-reported outcomes, and quality of life, which we will not be really talking too much about that area, but the first primary and secondary objectives. Next one. This is the demographic and baseline characteristics. So like I say, and maybe I didn't mention that before, this is a randomized placebo-controlled study, meaning some patients were placed into the placebo group where they are not getting the drug, and then some of them were in the drug group, which is Dalzota at the 5 milligram per kilogram. Remember, we're talking about B1, which is the lower dose. Here is just showing you the mean in terms of age. So patients were distributed, you know, we said the ages already, but they were around 13, 16 years of age. BMI is important as well when we do this kind of treatments. Age of onset of symptoms, it was balanced between the two groups, around 3.8, 3.6 years of age. Creatine kinase are high as a baseline. So these are the baseline numbers for these patients. So 7,000 for the placebo group, and then 5,000 for the treatment group. And corticosteroid use was required on everybody before and during the study. Next. Like I said, the first primary endpoint that we look in this case is safety and tolerability. And it's always important with every medication we try. It's important to see here that there were no related adverse events that occur in more than one participant, meaning adverse events that were associated to the drug. They were not really not related in this case. One participant discontinued due to a serious adverse event of anaphylaxis, which fully resolved. And one participant discontinued due to moderate infusion-related reactions. Moderate, we classified the reactions to the patients. So we have anaphylaxis that is more severe, moderate or mild reactions to the infusions. In this case, it was a moderate, and the person decided to move out of the study. There were no symptomatic hemoglobin changes or hypomagnesemia or renal events. Why is this important? Because we have had story in the past with other products where we had low levels of magnesium, for example, or we have problems with hemoglobin. So there were no changes in those. And to me, that was one of the first things that caught my attention, because it's extremely important. Less side effects that we have to deal with, obviously, always better. And there were no adverse events leading to that. Next. So the initial data, again, from the 5 milligram per kilogram cohort, this is what we're gonna look. And next. I will show you more in detail over the next slides about these points, but let me just say it in general. One of the first things that you look with these products, this is a monoclonal antibody that is connected to the PMO. The PMO will be the medication that will help doing the skipping in the cells. So one of the things that you want to look is that, is the medication really getting where it's supposed to be getting in the muscle? In this case, this drug is going to be given not as the ones we have already approved right now commercially for other exons, which is once a week. These are less administrations. So you really want to know that the medication is getting where it's supposed to be getting. And there was a consistent delivery of the PMO, because the monoclonal antibody will take it and it will release the PMO to the skeletal muscle. The second group, or the second part that we were looking at, is the exon skipping. So how much the patient is able to skip with this new drug. So 37% of mean increase in exon 44 skipping. I'll show you a graphic that it will be better to understand what I'm trying to say, but it was definitely, there was evidence of exon skipping for this specific drug. And there was up to 66% increase in exon 44 skipping, but that refers to specific patients. So the 30% is like a mean number for like the whole group. 66% is there was at least one participant who had even up to 66% of the exon skipping. The other thing we look at the dystrophin production, because it's important to see not only skipping, but how much dystrophin is the patient able to produce. The mean for the group was 25%. of the normal production of dystrophin. And again, the graphic will help us understand that better. And there was a participant who had up to 54% of the normal. So that's when, you know, when we explain this to families and when I talk to the families, I have to be very open. And to me, this is important because this is the highest level we have had in terms of expression of the dystrophin protein with this group of products, with the exon skipping products. And then the creatine kinase as a biomarker, which, you know, would usually me, I try to measure it at least once a year. And in this case, we see that the creatinine level actually decrease to almost normal levels, or around 80% of reduction compared to the baseline in the patients. And again, I'll show you that in a graphic much easier. Next one. So exon skipping, I asked Avidity to add this because sometimes it's hard to understand what is that we're talking. Like I said, there are exon skippings for other deletions. For this one, it's just specific group of patients where it does is that the PMO is attached to the monoclonal antibody. They bind to the receptors that are outside the cell. This structure gets inside the cells and gets inside the nuclei where it releases the drug. And what is that it does? For some specific patients, for example, in this case, if you're seeing here, you have 42, 43, 44, 45, 46. If you remove 44, or you really skip, I always say remove because I like to think in my mind that is removing, but it's really skipping that. You're kind of reconnecting 43 to 45, which again, for some patients, that is an ideal situation to be able to correct the defect that specific patient has. And what it's important, like I said before, is that we are going to require less frequent dosing. To me, I always tell my patients, it's also about quality of life. It's not only therapies, but also quality of life. And I think quality of life will improve definitely as we move forward with less infusions, not having to do once a week, but hopefully once every 4 weeks. Next one. So that's what I was explaining before, but I'll just say it really quick. Again, the monoclonal antibody connects to the, or is paired with the PMO, and this connects to the transferrin receptors, goes inside, release the drug, and hopefully we're trying to correct or skip the defect and start increasing the dystrophin protein. Next. When I mention the first part, when I show you the 4 little squares, the first one we talk about how much is getting to muscle. And like I said, these numbers not important for all of us, but it's important to know that there was enough medication, enough drug got to the muscle. So we know if you see the left side, it's in healthy volunteers, always only with a single dose. And then on the right side, we have patients that were on the trial with exon 44 skipping. They received the medication, and we know it was measured in the muscle, and it was definitely enough expression or enough medication that got to the muscle. Next one. This one it's important. I was saying 37% increase in exon skipping. So you see the two groups, the placebo group and the treated group. In the placebo group, you have a baseline and you have 4 months later. So there was no increase of exon skipping, obviously, because they are in the placebo group. But the ones that were treated with Dalzota, you see that there was a baseline, and after 4 months of treatment, there was a 37% increase over their baseline of exon skipping. So we know that the medication is doing what it's supposed to be doing, and it is a significant result. Then the next one. The next thing we look at the dystrophin production. Same thing on the left, you have the placebo group, which You don't have obviously dystrophin production because you're not treating them with anything. In the group that got treated, there was a baseline production that the patient has, but then 4 months later, they have up to 25% increase as a group. Again, this is as a whole group. But then there were participants that had restoration of the dystrophin up to 54% of normal. Dystrophin, why we look obviously dystrophin, because this is what we want to try to produce to try to make the cell work again fine and prevent the muscle destruction progressively. Next. And then creatine kinase. Creatine kinase tells about the health of the muscle. The creatine kinase, it's released every time there is breakage of that muscle, there is damage of that muscle, that creatine kinase goes to the blood. And that's why our patients have very high levels of creatine kinase. So it's important that if we're trying a product, measuring the creatine kinase will tell us that what's going on. There is hopefully less destruction because there is more production of dystrophin. So you see here the comparison of the group of the placebo, which will be the gray line. And we said that most people start around like 6,000, 7,000. And then you see the ones in blue on the lower part, where you see the numbers start going down and they got almost to normal levels. So extremely, extremely important. There was a reduction by greater than 80% compared to the baseline. Next one. And right now I'm gonna let Hussam Younis to talk about transforming the future of Duchenne together. Hopefully at the end, we'll have the section for question and answer if there is any doubt, that anything that we need to talk a little bit more about the science. But again, I'm very excited. Every step we take, it's a huge step for our patients. So thank you so much for letting me be part of this.
Thank you, Dr. Castro. My name is Hussam Younis. I'm the Global Program Head for the DMD44 program at Avidity Biosciences. Next slide. So as Dr. Castro nicely shared with you today, our preliminary data with the B1 5 mg per kg cohort. We also have a higher dose cohort, B2 cohort at 10 mg per kg, which at this point is fully enrolled. And we're really committed and focused to wrap up that portion of the study to develop a really robust data package that we really plan on moving forward with and interacting with regulatory agencies on how best to expeditiously move this program forward into accelerated approval. Our initial design had potentially going up to 20 mg per kg. At this point, we don't believe that we need to go up to 20 mg per kg. The 10 mg per kg will be the final cohort in this trial. As Dr. Castro mentioned, participants in the B1 and B2 cohorts have the opportunity to roll over into the EXPLORE44 OLE trial. At which point all the patients in that cohort will be on Dalzota active drug, which is very important. And also we have plans to further expand enrollment into the EXPLORE44 OLE up to 10 to 15 patients. Next slide. So Avidity, as Sarah started off, is really committed to partnering with the DMD community to efficiently advance treatment options for DMD and really understand and appreciate the urgency in which we need to do this. To that end, Avidity has applied for orphan drug designation that we were granted for Dalzota. Both in terms of fast track and rare pediatric disease designations as well, including orphan designation in Europe. We are committed to interacting with the FDA as quickly as possible to really think about the most expeditious way to get this drug to patients as soon as we can. Next slide. So I really want to thank everyone that has participated in our trial, including the patients, the caregivers, all of the investigators. Of course, this trial is still ongoing, so it's very important for us to focus on completing the EXPLORE44 trial, and then ensuring that we also complete the EXPLORE OLE trial. To that end, I really want to thank the DMD community on behalf of all of the scientists and drug developers at Avidity Biosciences. Sarah, I'll hand it off to you.
Thanks, Hussam. So just to close, in terms of, you saw a picture of the whole team at Avidity, and we're working incredibly hard around trying to deliver on our vision of profoundly improving people's lives by revolutionizing the delivery of RNA therapeutics. You have a whole team at Avidity who is committed to bring their best work, our best science, to be able to make an impact in people's lives. And with that, I think we're going to move into Q&A. So if everyone can turn their camera on, and Michael, I'm going to hand over to you to facilitate for us.
Thank you, Sarah. And let me begin. Congratulations on the data. It really is a very compelling package whenever one sees all of the movement across both skipping, the safety profile, the dystrophin production, and then certainly ending up with CK levels that are remarkably reduced. So I think the community, not that I think, I know the community is really focused on this program and the ones that are going to follow it. So I think it really is an exciting time for Avidity and this type of technology. Let me begin with a question around where we are today and what the company's thinking about. And it's really directed at you, Sarah.
Mm-hmm.
Is there plans to go for accelerated approval with the 5 mg dose? Are you going to look for the 10 milligram dose? Is there a strategy around here that you could share?
Yeah, so, our goal is to move as expeditiously as possible. First and foremost, we are looking forward to being able to work with the FDA and have a discussion with them around what is the most expeditious path to accelerated approval. Those conversations have not happened yet. The FDA is aware of the data, but there is a process by which you have meetings with the FDA. In relation to 5 milligram, firstly, the study is ongoing. So we're looking to understand in the fullest possible way the data from the EXPLORE44 study. In relation to the open label extension study, the plan is to roll 10 to 15 additional participants at both doses. So at this point, we're still very much exploring both of the doses and dose schedules.
Okay, thank you for that. And I'm also pretty sure that you're focused as an organization on the platform technology designation, because it seems that all of this approach using the antibody leans heavily into that and could really help accelerate future programs. Yes,
I think we're really excited and encouraged by some of the guidance coming out of the FDA with regards to platform technologies. And this is a platform technology. The antibody is the same. The linker systems are the same. And the chemistry is the same in DMD being PMO. And for our other programs, we use an siRNA. And again, it's the same type of chemistry, just different sequences. So certainly it's our hope that there's really good movement with regards to platform technology. And we can benefit from that given the consistency across our platform.
Excellent, thank you. A question for Dana and Hussam. Have you got plans in the near future for the open label extension patients to move to the 10 milligram dose?
I think I will let Hussam answer that one.
Yeah, so once patients complete the B2 EXPLORE44 portion of the study, they do have the opportunity to go into the OLE, including at the 10 mg per kg dose level.
Okay. And I know that we're going to be expecting functional data from the 5 mg dose. Is there any guidance you want to give the community and when we might expect to see some of that?
Yeah, Hassan, do you want me to take that?
Yeah, please.
In terms of, so the study is still ongoing. The data that we looked at was a 4-month time point. And, we very much look forward to sharing the data and plan to do that. It'll probably be next year and our idea would be to share it at a medical conference.
Okay,
But
thank you.
yes, absolutely, more data to come. As we have, people have been receiving drug for longer, the plan would be to look at the functional data as well.
Okay.
I wanted to make a clarification. Something that I didn't mention is that the patients got the biopsy baseline, and then the second biopsy was 4 months later. So this is the data we have in the biopsies 4 months. I don't think I mentioned that, and I think it's important.
Okay, thank you. Another question that I have is really, have you thought about, given the effect here and what you're going to learn out of the 10 milligram group, do you understand the PD enough that this is going to be once every 6 weeks, once every 2 months? Is there plans to investigate what an optimum dosing regimen may actually look like?
Yeah, Hussam, do you want to start with that one?
Yeah, that's a really good question. So the 5 mg per kg cohort we're currently studying every 6 weeks. And at this point, based on this preliminary data, very, very encouraging result in terms of that exon skipping as well as the production of dystrophin. And the 10 mg per kg is being tested at every 8 weeks. And so we're really committed, to complete the EXPLORE44 trial. And then at that point, really collect all of the data that we have to really make a data-driven decision on what is the most appropriate dose as well as the regimen. Because we really need to look at, the entire data package that we would have at that point, which we just don't have right now.
Excellent, thank you for that. Let me take one from the audience. Essentially, I picked this one simply because it's one that we've talked about before. And the consideration of patients that have previously received gene therapy, is that an option going forward? Is it in the company conscience somewhere?
Yeah, I think in terms of from a biological perspective, there's no reason why the two therapies could not be used. Either one before the other or one after the other. I don't think we see anything from a biology perspective that would create an issue there. Hussam, I don't know if there's anything you would like to add.
Yeah, no, exactly, Sarah. I mean, I think we've thought about the gene therapy and the utility of potentially having individuals be on both, and from a biological perspective, we don't really see any issues with that.
Erica, do you want to bring up some questions that you've got on your side?
Yes, I'm actually looking through. 1 second.
Yeah, I tend to get carried away whenever I get the microphone.
Erica, I answer one privately already. Somebody asked if we're going to do this in Spanish, and yes, we are. We're working on it.
Correct. Thank you, Dr. Castro. Yes, I was going to mention that we're going to work with the Yakari Foundation to translate this presentation and have a Spanish-speaking webinar as soon as we can arrange with Dr. Castro's schedule as well. One of the questions that has come up is just treating other exons, so exon 45 and the other exons. What is the thinking around that for Avidity?
Yeah, we have an exon 45 program that is in what's called the IND enabling phase. So that is still in a preclinical phase of development. IND is initial new drug. So when we have completed that phase, we then do a package to the FDA to request to proceed with clinical trials in humans. So that program is still in that phase. We also have earlier in our lab, we have other exons that we have also looked at skipping and sequences. And what we'll be working through now is the scientific process of studying them conjugated to an antibody and how that looks.
Perfect. Thanks, Sarah. I actually have two questions for Dr. Castro. So Dr. Castro, one of them is, what is the level of dystrophin levels with Dalzota compared to dystrophin levels in healthy individuals? And the second one is, what is the normal levels of creatinine?
Yeah, I love that question. I almost answered it all myself. Yeah, so the normal, every lab will report a different normal level, but it's usually around 250 to 300. In neuromuscular medicine, we accept 300 as a normal level of creatine kinase. And I always like to tell that the patients to compare of what we're talking about is 7,000 in these cases. For example, most of patients, 6,000, 7,000. So very, very high levels. And I think somebody was asking, so what I said, 37%, no, the reduction, I'm sorry. Remind me for the reduction of the creatine kinase at the end. We say 80%.
80%. 80.
So thank you. So yeah, I say 80%, but it's important. You see where the line starts, the blue line, we're talking about 4,000, 5,000, and you see the number falling below 2,000 and touching where it says upper limit of normal, meaning we're talking about 400s, 500s, and that's usually what we will consider kind of, on the lowest part. So that is what I was trying to say about the CK level. Was there a question about the expression of dystrophin as well?
Yeah, the question was, what is the level of dystrophin levels with Dalzota compared to dystrophin levels in healthy individuals?
Exactly. So we have the data here for the patients that were included. So the exon 44 amenable patients. And what I tried to say, the 25% was because in this population, if you see the graphic on the right side, patients were on average around 7% expression of dystrophin. And then they got up to around 32%. So there was a difference. There was an increase in dystrophin around 25%. So that was kind of the difference between where they were before baseline and then where they were 4 months after in the muscle biopsy, right? This is all measured on the muscle biopsy.
Great. Thank you, Dr. Castro.
Of course.
Michael, I'll turn it back to you if you have any other questions.
Yeah, the obvious one that I've seen on a couple of times, outside of the US, what's the plans for the European theaters, for example?
Yeah, so in terms of studies, the studies are ongoing. Our next step is to have a discussion with the FDA around what's the most expeditious path to accelerated approval. In Europe, the accelerated approval path, there isn't an equivalent. Our commitment very much is to develop our drugs globally. So we will be looking forward to having discussions with regulators in Europe around what we will need to do from a trial perspective there as well. And then typically sort of that European trial approach that we'll also look at most likely other sites as well around the world. Hassan, I don't know if there's anything you would add there.
And the timing
Nothing
for
else
the
to
10 mg? Oh, I'm sorry. Go ahead.
Yeah, nothing else to add.
Okay. And the timing for the 10 milligram dose?
So that data, EXPLORE44 is ongoing. We would anticipate that the next readout from the study we'd look to present at a medical meeting. That's probably going to be 2025.
Okay.
So I think in terms of for this year that the study's got to continue through, and then as soon as we have that data, we'll then look to share it. The ideal would be to present at a medical meeting. I know for Dr. Castro that's really sort of that it's scientifically that's very important and important to share with the community as well. So if we can time it with a medical meeting, we will.
MDA next year.
Yeah.
Eric,
Michael,
exactly.
may
Yeah,
I bring one point that I know you're going to explain much better than me? For these specific patients that are amenable to exon 44 skipping, one of the questions is why they have higher levels of dystrophy. And I
yeah,
think that's a big question.
yeah. So it's one of the challenges that's really affected companies moving into 44 because there is slightly more revertant fibers typically in this type of patient. People will euphemistically say 44 is easier to skip. It's not. There's other things that are going on in there. But in these individuals, we typically see a slightly milder disease because on average, you'll see 3, 4, 5% of revertant fibers. I think part of the reason for that is that intron for 44 is exceptionally large. It's larger than every other exon in the gene. And so it takes a long time for that intron to be read. And that there gives time for exon skipping drugs to do the job. And that's why I think this is so efficient.
And revertant fibers is just the fact that they kind of autocorrect
Yeah,
naturally.
they do naturally skip. And so they naturally skip to the extent of about 3 to 5%. And you've picked up roughly about 6 to 7% of them there.
Exactly. Okay, thank you so much.
Yeah, and one other thing I should say I'll add in here, and it's really a tremendous credit to Avidity. Many other companies have shied away from 44 as a target because you do have revertant fibers. And the only way really to show an effect is to have a pronounced effect. And it really speaks to the power of Dalzota, where you're now seeing it move from 5 or 6% up to 30-something%. And that there really has unlocked the key to targeting exon 44.
Yeah. It's interesting, Michael. I remember when we started with this program and some people said to us, you guys are crazy.
Yeah.
Why are you looking at that? Because it's going to be so hard. And part of our orientation, when I talk about our vision, we're looking to make a profound improvement in people's lives. And we specifically started with 44 because that's where others weren't going.
Yeah, and I believe that too at the time, Sarah. I think that if you were targeting 44 with an exon skipping approach that was going to put in 1 or 2% dystrophin, you would never see it above the background. And you really needed something that was going to target muscle and deliver something right into the cell. And there's why this works so well. And it was really made for exon skipping. Erica, have you any other questions on your side?
There's just one question on just the adverse reactions and understanding a little bit more on the adverse reaction and what may have caused that.
Sam, do you want to take that?
Yeah, I'll take that. The anaphylaxis reaction, essentially this is kind of an idiosyncratic reaction that happens not uncommonly with biologics. As Dr. Castro mentioned, the molecule here is an antibody conjugated to oligonucleotides, so it may be a reaction to the antibody. We have not seen this type of reaction across the platform. As Sarah mentioned, for all of our other molecules that we have, it's the same antibody, the same linker. It's just that the oligonucleotide is different. So it's not something that we've seen. This is the only event that we've seen it with at this point.
Thank you, Sam.
Okay. Any more on your side, Erica, before I start to wind things down?
No, I think we've answered most of them that have been on the chat. So no, I'm good on my end.
I will say I've seen a few of them that I just held back and thought that we'll give written answers to them over the next few days. I think that this program really speaks for itself with the data that's out there. I want to thank everybody for joining the webinar this morning, and thanks to Dr. Castro and the whole team at Avidity Bio for taking time out of what is a very busy day to share this data with the community. We certainly look forward to further updates and further developments on it. A post-webinar email will be sent out tomorrow, and we'll let you know when the webinar is going to be posted online for viewing or sharing with the community. And it just remains for me to thank everybody for joining us today and have a great rest of your day.
Thanks, everyone.
Thank you. Bye-bye.
Thank you.